Nick Curto. He is going to talk to you about our Seagrass Nursery and the work that we're doing growing seagrass. Nick has been here since before I was here, and so there are aspects of our Seagrass Nursery that are that he knows all about even better than I do, and so he's just going to introduce some of the work that we're doing.
Thank you for that wonderful introduction. So, just to kind of highlight, this is kind of a shortened version of a presentation I did about a couple weeks ago. It's already been uploaded to the Marine Resources Council YouTube page. So, if you kind of want the full version of this presentation, highly recommend checking that out. So, jumping right into Florida Oceanographic Seagrass Nurseries as a whole, we're going to kind of go through a brief overview of seagrasses. Yes, I know many of you are familiar with this knowledge already, but seagrasses are what are important foundation species in the aquatic environments around this area. They form habitat for other species, especially those in their juvenile life stages, and they are a primary food source, especially for those wonderful charismatic megafauna that we all love and hold dear, like our manatees and sea turtles. Another important aspect of them is they also influence the chemistry of the environment in the form of oxygenating the water and changing the sediment composition as a whole. So there are many different species of seagrasses found throughout Florida. The three that we focus on primarily with us is Seagrass Nurseries or manatee grass, Cauduli writing or shoal grass, and Thalassia testudium or turtle grass. And I always include these images because I always like people to actually take a moment to look at them, so they're able to kind of see like the different physiological differences between them. Like you can see like our manatee grass kind of looks longer and is the best way to describe it, more cylindrical. Our shoal grass tends to be more that wispy variety that's like traditional grass, and then our turtle grass is definitely the thicker, broader leaves that are more distinct out of the species. So with seagrasses, the unfortunate thing that we have to talk about as well is anthropogenic influences that oftentimes can devastate complete meadows or wipe them out completely. And this happens oftentimes in the form of freshwater discharges, algae blooms, and poor water quality. And I love this next slide because this highlights like a night and day difference of that and a perfect example. So this is back in 2016, actually just south of us from the Stork Causeway Bridge. So this area highlighted in the circle, full of seagrass. That's all those dark patches that you see. Fast forward to 2017, it's all gone. And that was due to the fact that there was a large discharge event that occurred in this time frame and basically wiped out all the grass in this area. So not only can discharge events harm seagrass, but also harm oysters, game fish, and other species that do not move out of those areas after a certain amount of time. So the important role in nurseries as a whole is to establish stock of various seagrass species for research and restoration purposes. And to kind of like jump into an aspect of what we're doing. So we have to focus on the past first. So for those who have not been here, and who also predate me, some of you in the audience are familiar with this part of it, is volunteers and staff would actually go out along the shorelines and collect viable fragments of seagrass. This is actually how the good chunk of our seagrass in our nursery originated from. However, one of the big things that we were trying to figure out is, yeah, there's a high potential genetic diversity, but where did it all come from? We didn't necessarily know this for a long period of time. So to answer this question in the form of manatee grass, we worked with Dr. Seagrass Garcia and Dr. McCoy at HBOI, who's in the audience, in a project funded by MOAT, and we took samples of manatee grass from all these different areas, as you can see, all the way down from the Keys to the northern part of the IRL. So a pretty broad area to collect samples from. And so yes, big scary image, I know. This is a dendrogram, and to kind of break it down with all the different colors and lines and everything, groups of individuals are collected from the different populations that we see represented by the key down below there. So the horizontal lines indicate the distance between connected groups, which is represented by the bar right there. And to better highlight that as well, I circled the two different groups that are kind of distinguished by the dendrogram, where you can see on the bigger circle, those individuals are more in common with one another than the other ones in the smaller circle. The vertical lines indicate similarities, so highlighted by this and this here. So the individuals in the left bar there are similar compared to those ones which are further down on the dendrogram, which they're not as similar as those ones. So we can tell where these plants were from and how closely related they are to one another. So to highlight this, I'll focus specifically on Florida Oceanographic Society and Harbor Branches Nursery. So Harbor Branch is represented by the blue circles indicated by the black arrow there, and you can see they're all kind of clustered together for the most part of the dendrogram. However, with Florida Oceanographic, you can see there's some clustered at the bottom of that one, there's some clustered there, there's a group that's all by itself over here that's not related to any of the other locations, and then there's a final one up there. So you can see like the circles that are with them as well, they're all from different locations, whether it's Loxahatchee, Lake Worth, JC. These are different areas and different populations of grass in different systems. So what are the implications of this? FOS has genotypes from different regions of the IRL. So kind of what we figured out before, but this basically solidifies this. So the important thing about this is they might be locally adapted to different stressors, whether that's variations in salinity or temperature or natural light like turbidity. And so this opens the door to potential future experiments, which I'll talk about in a little bit. So, but the other important thing is it helps us with informed restoration as well. So this can kind of increase the diversity of our restoration plots by being able to actually pick and choose certain ones from certain areas, or maintaining locally adapted genotypes. Like for example, the ones from the Loxahatchee stay in the Loxahatchee, or the ones from Lake Worth stay in the Lake Worth Lagoon. So it just depends on what we wish to do with this going forward. So with the sequencing of the native grass, it's allowing us to understand the differences between these populations. But in addition to that, we've also sequenced the genome of how really ready, which we were the first to do. And we've also done that sequence data that compares the grass to a mass mortality event back to the source population in our nursery. And to better represent this, I know, another dendrogram. A little more simpler than the other one though. So all these samples were originally from the meadow space. And so you can see it's represented by 3 colors, canes, FOS, and the meadow. And so this is from a project we did a couple of years ago where we took grass from our meadow and planted it in 2 different locations. All that grass died in the majority of those locations, and we actually went back months later to actually sample the surviving grass. And so you can see certain grasses from canes are closely related to samples from the meadow, certain samples from FOS are the same as well, but then there's other distinct ones that aren't related to anything in the meadow, which is really interesting because that just shows that even the meadow space is very highly genetically diverse. And so to kind of break that down a little more, we're jumping into now a kind of transitional phase in our front of house space, which we're making more into a genetic library. And so to represent that, we actually have several tanks now that are kind of genetics tanks, if you will, where we actually have those certain fragments or certain variants that you saw in the previously represented dendrogram, and they're now in their own separate tanks represented by those blue tags there. So we're able to keep track of which ones are which, and we also have barriers separating them as well. So for example, in the image on the right here, those are actually 2 different genetic variants in the same tank. So the exciting thing about this is, I don't know if anyone else has been in the back of our house recently, but this is our back of house experimental array. And the idea is each of these 4 groups of tanks can actually be on their own separate unit. So we can actually run different experiments with them. So the idea is with these genetic variants, we can actually run experiments with temperature, with salinity, with turbidity, hopefully, and do those stress testers that we were talking about, and hopefully open the door to finding different variants that work better in certain conditions compared to others. And so, to wrap it up all nicely, I wanted to show a wonderful picture of our meadow space and open the floor to any questions.